I want you to imagine a virus that is so unbelievably contagious that just a single infected person breathing in a room can spread it to like 18 other people.
Right, yeah.
In the epidemiology world, they call that an R naught of 12 to 18, which is just a staggering number. But here is the truly terrifying part of this.
Oh, the immune amnesia.
Yes. This virus possesses the ability to cause immune amnesia. It literally wipes out your body's memory of how to fight off other diseases you've already beaten.
It really is biological sabotage on just a massive scale. I mean, for a long time, it was one of the leading causes of death among children globally. We are, of course, talking about the measles virus, or MAV.
Right. Now, thanks to the live attenuated vaccine, most of us have been protected for decades. But the crazy thing is, despite having a vaccine, we've never truly understood how human antibodies actually neutralize this virus on a molecular atomic level.
Exactly. We've mostly relied on mouse models, to guess how our immune system fights it.
That is, until now. Today on this deep dive, we are looking at a really groundbreaking 2026 study by Acciani and colleagues. And this was published in Cell Host and Microbe.
Yeah, it's a fantastic paper.
They didn't just guess with mice anymore. They decoded the exact, precise molecular locks our bodies build to shut measles down.
What's fascinating here is that shift from animal models to a fully human blueprint. We are finally looking at the exact molecular mechanisms of human immunity against measles, mapping it literally atom by atom.
Okay, let's unpack this, because we're going to explore the viral machinery, the discovery of this amazing super responder donor, and the real-world clinical implications. But if we're going to understand how the body stops this virus, we first need to understand how the virus attacks. So how does measles actually breach our cells?
So it relies on two essential surface glycoproteins. You can think of them as the virus's specialized entry tools.
Okay.
The first is hemagglutinin, or just the H protein. This is a transmembrane protein that groups together into dimers or tetramers.
Wait, let me stop you there for a second to make sure I'm following and that the listener is following too. Transmembrane, meaning it sticks through the virus's outer layer.
Yes, exactly. It protrudes from the viral envelope, and its entire job is to find and bind to specific cellular receptors on your cells.
Like finding a very specific doorknob.
That's a great way to put it. And depending on the cell type, it targets different doorknobs. So on your immune cells, it binds to the SLAMF1 receptor. On your respiratory cells, it targets Nectin-4.
Oh, wow.
And just for completeness, in lab-adapted strains, it can also use CD46. But essentially, yes, the H protein is purely the grabbing mechanism.
Right.
But once it grabs hold of that doorknob, the second tool goes to work, and that's the fusion, or F protein. The F protein is what virologists call a metastable class I trimer. It's composed of these F1 and F2 subunits.
Okay, hold on. Metastable. That makes it sound like it's, I don't know, incredibly fragile or barely holding itself together.
It is a really crucial word here. It means the protein is locked in this high-energy, spring-loaded state, just waiting for a trigger. It is under immense physical tension.
Like a mousetrap.
Exactly like a mousetrap. When the H protein successfully engages a receptor, when it grabs that doorknob, it sends a chemical signal to the F protein.
And then what happens?
The F protein undergoes this massive, violent structural refolding. It essentially shoots a fusion peptide right into the host cell's membrane.
Jeez.
Yeah, and then it forcefully collapses into a stable 6-helix bundle. That physical collapse pulls the virus and the human cell together, creating a fusion pore. It literally bridges the two cells so the viral payload can spill inside.
Okay, to help you visualize this at home, I want you to imagine a pirate ship attacking a massive galleon. The H protein, that's the grappling hook, it's thrown over to lock onto the target ship's railing. The F protein, on the other hand, is like a spring-loaded boarding plank combined with a battering ram.
Yes. And to take your pirate ship analogy a step further, imagine if that battering ram was under so much physical tension that the moment the grappling hook clicked into place, the ram just snapped forward automatically, smashing through the hull.
That is a terrifying visual for a virus. Okay, so let's do a quick recap here. The H protein is the grappling hook for receptor binding. The F protein is the battering ram for membrane fusion.
Correct.
So if you're listening to this, I want you to ask yourself, if you were a microscopic defender trying to stop this invasion, would you rather cut the grappling hook or jam the battering ram? Keep your answer in mind as we see what the human body actually does.
Well, to figure out what the human body does, the researchers had to find the right human to study.
Right. Which brings us to the protagonist of our deep dive today. A person identified in the study simply as donor number 3920.
Yes, donor 3920.
So how do we actually figure out what a human body does here? Did they just pull blood from a random person and hope for the best?
Not quite random, no. They screened 20 different human donors in La Jolla, California. They were looking for individuals with really strong immune responses. And they found this incredible outlier.
Our donor 3920.
Right. She is a 56-year-old female who was vaccinated in childhood and received a booster about 5 years ago. And when they checked her blood, she had these massive, off-the-charts, circulating antibody levels against measles.
But when they looked closer at her blood, using flow cytometry, they found something that completely flipped the script, right? Something that contradicted what scientists had assumed for decades.
It really did. So flow cytometry is a way to sort and count individual immune cells using lasers. And using this, the researchers discovered she had roughly 3 times more memory B cells specific to the F protein than the H protein.
Which is wild, because for years the assumption was always that neutralizing antibodies primarily targeted the H protein, the grappling hook.
Exactly.
They thought, if you block the virus from grabbing on, you win. But her body was overwhelmingly focused on targeting the battering ram instead.
Her immune system essentially decided that the fusion machinery was the much bigger threat. But that realization created a massive technical hurdle for the researchers.
Oh, because how do you actually get those specific antibodies out?
Right. How do you pull those highly effective antibodies out of millions of cells to see exactly how they work?
You can't exactly use tiny tweezers.
No, you can't. So they used a cutting-edge piece of technology called the Bruker Cell Analysis Beacon. They took her activated memory B cells and loaded them into what's called an 11K nanopen optochip.
Okay, I have to jump in because I read this part of the study and I'm just obsessed with it. Nanopens and optochips. They are literally using microscopic cages made of light, right? Optofluidics.
It sounds totally like science fiction, but yes. Optofluidics uses light to create physical pressure. This light pressure guides and traps individual living B cells into microscopic pens on the chip. There are 11,000 of these little pens.
That is unbelievable.
It's amazing tech. Through this single-cell microfluidics technique, they were able to isolate the exact cells producing these incredible antibodies. Then, they sequenced their unique genetic codes and cloned 384 of them into a line of lab cells called XBCHO cells.
XBCHOs.
Yeah, they're basically a standard mammalian cell line used in labs to mass-produce proteins. So by cloning the genes into these cells, they could manufacture the antibodies in large enough quantities for testing.
Got it. And what was the final yield from all of that?
In the end, they successfully isolated 52 monoclonal antibodies against the H protein and 46 against the F protein.
Okay, so let's do another quick recap to make sure we are all tracking the story so far.
Sure.
They screened 20 donors. They picked donor 3920 because she had sky-high F reactivity. Then they used literal cages of light nanopen tech to isolate roughly 100 highly specific antibodies from her blood.
That's the summary, yeah.
So to the listener, I ask you, what stands out to you about the fact that her body dedicated 3 times more resources to targeting the virus's battering ram rather than its grappling hook? It really flips our understanding of immunity. But anyway, let's look at how these weapons actually work. Let's start with the grappling hook cutters.
Yes, the 52 monoclonal antibodies, or MABs, targeting the H protein. The researchers binned these into 4 major epitope clusters. They named them HE1 through HE4.
An epitope is just the specific part of the virus the antibody grabs onto, right?
Exactly. And to map these, they used high-throughput surface plasmon resonance.
No, surface plasmon what? That sounds incredibly complicated.
It is a bit of a mouthful, high-throughput SPR. Essentially, it's an optical technique where you bounce light off a surface to measure in real time exactly how and how tightly 2 molecules stick together.
Okay.
It tells them precisely which part of the viral protein the antibody is grabbing, and then they paired that SPR data with cryo-electron microscopy.
Cryo-EM, which is essentially flash freezing the virus and taking atomic-level photographs of it.
Right.
So when they developed those atomic photos, what did they see?
They found that a massive 81% of these H-targeting antibodies all swarm the exact same spot, the HE1 cluster.
And where is HE1?
It is the apical surface, the very top of the H protein. It perfectly overlaps with the sites where the virus tries to bind to our cellular receptors.
Ah, so they're just swarming the tip of the grappling hook, so it physically cannot find a rail to grab.
Precisely. And there are 2 specific antibodies in this HE1 group that the study highlights in great detail, 4D08 and 1CO2. Let's talk about the first one, 4D08.
Okay, 4D08, what's its strategy?
What's fascinating here is how 4008 works. It targets a subcluster called HE1B. 4008 uses a flattened loop in its molecular structure, specifically in its CDRH3 region, to perfectly mimic the shape of the human SLAM-F1 receptor.
Wait, it wears a disguise.
Yes. It tricks the virus into grabbing the antibody instead of a real immune cell. And when 4008 binds, it causes what we call a register shift.
A register shift. What does that mean in this context?
It means it physically pulls the beta sheet of the viral protein outward by 3.5 angstroms.
3.5 angstroms. Okay, for the listener, an angstrom is less than a 10 billionth of a meter. It sounds tiny.
It does, but in the atomic world, moving a structural protein by 3.5 angstroms is a massive remodeling. It's an earthquake.
It essentially warps the grappling hook so badly it can't function. Wow.
Nah.
But what about the other one, 1CO2? You said 81% swarm the top, so is 1CO2 doing the same thing?
Well, 1CO2 targets an adjacent spot, HE1A, and it also causes that register shift, bending the protein. But here's the catch. In live animal models, 1CO2 is far less protective than 4008.
Really? Why, if they are both bending the hook?
Because if we connect this to the bigger picture, we have to look beyond just the physical blocking action and look at the antibody's tail, the FC region.
Right, because antibodies don't just act like physical shields, they also act like signal flares. The FC region is the part that calls in the immune system's heavy cleanup crew.
Exactly. 4008 triggers high levels of a process called ADCD, which stands for antibody-dependent complement deposition.
ADCD.
Right. It is incredibly good at calling in immune cells, like macrophages, to swallow and destroy the virus once it's tagged. 1CO2, despite binding well physically, does not trigger this cleanup response effectively.
Okay, that makes sense. But I'm looking at your notes here on another antibody, 1CO8, and I genuinely want to push back on this because I don't get it.
Okay, let's hear it.
You just said the main strategy for the H protein is blocking the very top where the receptor is, right? The HE1 cluster.
That's where 81% of them go, yes.
But this 1CO8 antibody attaches to cluster HE4, which is on the underside periphery of the H dimer. It completely, entirely misses the receptor binding site.
It does.
So wait, if 1CO8 doesn't even block the receptor, how on earth is it neutralizing the virus with picomolar potency? How is attaching to the bottom doing anything useful?
That is the beauty of molecular mechanics. Remember earlier when we talked about how the virus machinery works? The H protein doesn't work alone. It has to physically and chemically communicate with the F protein to tell it to deploy the battering ram. Yes. By anchoring itself to the underside periphery, 1CO8 acts as a structural wedge. It likely prevents the H protein from communicating with the F protein.
So it hits the blind spot. It stops the invasion sequence before the battering ram can even get the signal to trigger.
Exactly. It's a highly potent strategy, and the region it targets is over 99% genetically conserved across different measles strains.
Okay, let's recap this section. We have 4 H clusters. Antibody 4D08 blocks the receptor by acting as a decoy and triggers immune cleanup. But antibody 1CO8 attacks the virus's blind spot to stop the H and F proteins from talking to each other. And both targets are over 99% conserved.
Spot on.
So listener, think about 1CO8 for a second. Isn't it fascinating that attacking a virus's blind spot works just as well, if not better, than hitting it head on?
It really is an elegant solution.
So we've got the grappling hook covered. But as you mentioned earlier, donor 3920 dedicated the vast majority of her immune resources to the F protein, the battering ram.
Yes, the F protein.
Here's where it gets really interesting. How did they map the F protein antibodies?
Similar to the H protein, the researchers mapped the F protein antibodies into 5 distinct clusters, which they called FE1 through FE5. And they mapped these vertically, starting from the base of the F trimer all the way up to its apex.
Okay.
And when they looked at the data, a very clear biological rule emerged. Location is everything.
Meaning some spots on the battering ram are better to hit than others?
Much better. Antibodies that hit the bottom half of the F protein, so that's clusters FE1 and FE2, those are surprisingly weak neutralizers.
Interesting.
But the antibodies that strike the top half and the very apex FE3, FE4, and FE5, those are incredibly potent.
Okay, so let's talk about the apex predators of this group. The study focuses heavily on 2 specific F antibodies, 4FO9 and 3A12.
Right. So 4FO9 hits cluster FE5, which is the absolute peak, the apex of the trimer, and it has this specialized feature, an 8-residue tyrosine-rich loop.
An 8-residue tyrosine-rich loop. What does that let it do?
It acts almost like a claw. It allows the antibody to reach out and grab 2 different sections, or protomers, of the virus protein simultaneously. It binds them together.
Wow. And what about the other one, 3A12?
3A12 binds just a bit lower at cluster Fe4, that's the upper center, and it wraps itself across both the F1 and F2 subunits at the same time.
But, okay, if this F protein is a high-tension spring, how do the scientists actually prove that these specific antibodies are the things stopping it from firing? Do they, like, poke it with a microscopic stick?
Red a stick, no. They use heat. They use a technique called nano-DSF.
Nano-DSF.
Yes, it stands for nanoscale differential scanning fluorimetry.
Okay, scientists really do come up with the most complicated acronyms for essentially saying, we microwaved a virus, because that's what they're doing, right? They use lasers to heat up the protein until it melts and springs open.
That is a hilariously accurate way to put it. Yes, they apply thermal stress to see how much heat it takes to make the protein pop into its post-fusion state. Normally, the F protein springs open at a very specific baseline temperature. But when they attached antibody 3A12, it increased the protein's melting temperature by a massive 23 degrees Celsius.
23 degrees.
Yeah. And antibody 4F09 increased it by 19 degrees Celsius.
So to put that in perspective, I want you to picture taking industrial-strength duct tape and wrapping it incredibly tightly around a loaded, highly tensioned mousetrap spring.
That's exactly it.
No matter how hard that spring wants to snap, the tape holds it in its locked prefusion state. That is what these antibodies do. They mechanically lock the F protein so it physically cannot fire.
It is the ultimate atomic duct tape. In fact, remember how we talked about the Fc effector cleanup functions with the H antibodies?
Yeah, the signal flares.
Well, 4F09 has almost zero of those Fc effector cleanup functions, like ADCC or ADCP. It basically doesn't trigger them.
Wait, really? So it doesn't call in the cleanup crew at all?
It doesn't need to. It protects the body purely through sheer mechanical neutralization. The duct tape is so strong, the virus is just rendered completely inert.
That is mind-blowing. Let's do a quick bullet recap for the listeners. We have 5 F clusters. The top binders are the best. Antibodies 4F09 and 3A12 neutralize the virus by thermally and mechanically locking the F protein so it literally cannot fire.
That covers it.
So listener, think about this. If you could engineer a drug, would you rather trick a viral receptor or physically glue its molecular machinery shut? It's pretty clear the duct tape method is incredibly effective.
It is.
But, okay, so these antibodies are structural marvels in a petri dish or under a laser. But if you're listening, you're probably asking the obvious question here. Does this actually work in a living, breathing body?
It's the most important question. And to answer that, the researchers used the cotton rat model.
Why cotton rats specifically? Why not standard lab mice?
This is a critical detail. Cotton rats are one of the very few animal models that naturally replicate human SLAM F1 measles infection.
Because of this specific receptor.
Exactly. The virus behaves in their respiratory tract almost exactly as it does in a human's, so it's a highly accurate proxy.
So they ran 2 types of tests on these rats, right? Prophylactic and therapeutic.
Correct.
Prophylactic meaning they gave the rats the antibodies before they were exposed to the virus, and therapeutic meaning they gave them the antibodies after the infection had already started.
Yes. So let's look at the prophylactic trials first. They gave the antibodies 14 hours before exposure, and antibody 4F09, the one that glues the apex shut, was an absolute superstar.
What were the numbers?
It reduced viral titers in the lungs to entirely undetectable levels. It completely, 100% prevented the infection from taking hold.
That's amazing. But the therapeutic trials are where my jaw actually dropped reading this study.
Yeah, they gave antibody 3A12 to subjects 24 hours and in some cases even 48 hours after they were infected with measles.
Which is a lifetime for a virus that replicates that fast.
It is, and even with a 2-day head start, 3A12 reduced the viral lung titers by 500-fold. It stopped an active, raging measles infection right in its tracks.
But wait, let me play devil's advocate here. If there's one thing we've all learned over the last few years of living through pandemics, it's that viruses mutate. Variants are a huge problem in virology.
They absolutely are.
Did the researchers test these antibodies against different, currently circulating strains of measles, or just a lab strain?
They did test them against real-world variants. They tested these fully human monoclonal antibodies against the currently circulating wild-type strains. Specifically, they looked at genotypes B3 and D8.
Okay.
And they also tested the standard Schwartz vaccine strain. And here is perhaps the best news of the entire study. They maintained their picomolar neutralization potency across the board.
Universally. How is that even possible? Wouldn't the virus just mutate to avoid the antibodies eventually?
You would think so, but no. And the reason comes down to where these antibodies bind. because these specific antibodies target core structural mechanics like the exact underside wedge of the H protein for 1CO8 or the absolute apex of the F protein for 4FO9, those binding sites are incredibly stable.
Because the virus needs them to function.
Exactly. The epitopes for 1CO8 and 4FO9 are 99.4% to 100% genetically conserved across thousands of sequenced measles strains.
Meaning the virus literally cannot afford to mutate those specific spots, or its machinery just stops working entirely. It would be an evolutionary dead end for the virus to change shape there.
Right. None of these contact sites are known as positive selection sites for viral evolution. The virus isn't trying to mutate them, so the antibodies remain perfectly effective no matter how the rest of the virus tries to drift.
So what does this all mean? We have these ultra-potent, fully human-derived antibodies that act as universal atomic locks. How do we actually use this knowledge in the real world?
Well, if we connect this to the bigger picture, measles cases are unfortunately rising globally right now. This is largely due to gaps in vaccine coverage.
Right.
And while the live attenuated vaccine remains our absolute best primary defense, nothing beats preventing the infection in the first place. These fully human monoclonal antibodies could be deployed as a highly effective therapeutic tool.
Like a compassionate use cocktail.
Exactly. Very similar to how we use the drug Nursurvive for RSV in infants, this antibody cocktail could be given to people who are immunocompromised and can't safely receive the live vaccine.
Well, that makes sense.
Or to infants who are too young to be vaccinated, but are suddenly exposed during an outbreak, say, in a daycare center.
Let's recap this final segment real quick. These antibodies work beautifully in live animal models. 4FO9 totally prevents infection, and 3A12 treats an active infection even 48 hours later. And they have universal efficacy because those target sites are over 99% genetically conserved.
Perfect summary.
So, listener, ask yourself, how might a post-exposure cocktail of these exact antibodies change the way we manage measles outbreaks in elementary schools or hospitals? It could be completely game-changing.
It could. It would prevent the immediate severe respiratory symptoms, and more importantly, it could prevent long-term fatal complications like SSPE.
Remind me, SSPE is?
Subacute sclerosing panencephalitis. It is a devastating, fatal neurological condition that can occur years after an infant is initially infected with measles. Having a therapeutic like this entirely shifts our public health response from purely preventative to actively therapeutic. We could shut down outbreaks the moment they are detected.
We have covered a massive amount of ground today. And what really blows my mind is that this all started with a simple blood draw from a 56-year-old woman in California.
Donor 3920.
Right. From that single sample, using cages made of light and atomic-level photography and microwaving viral proteins, we uncovered the atomic duct tape and the grappling hook cutters that her immune system naturally engineered, all to perfectly counter one of the most contagious pathogens on Earth. It is a masterclass in biological engineering.
It truly highlights the elegance and the raw power of the human adaptive immune system. When given the right blueprint, in this case, from a standard childhood vaccine, the body can design molecular countermeasures with a precision that our best human technology is only just now learning to copy.
Which leaves me with a final provocative thought for you, the listener, to mull over as you go about your day. If one single individual's immune system, exposed to nothing more than a routine childhood vaccine decades ago, can naturally engineer such flawless, highly conserved atomic-level locks against a terrifying virus, what other microscopic miracles are currently floating around in your own bloodstream, just waiting to be discovered?